our earlier episodes, we have been talking about different methods of plant tissue culture, general requirements for plant tissue culture, and various applications of plant tissue culture. Now in this episode, we come to a more vital aspect, and that is practical problems encountered during in vitro cultures. These problems that we are going to discuss now are of general natures, but we would be mentioning one or two specific problems. I've got a group of researchers working in the field of plant tissue culture here, and I would like to hear from them the problems that they have been facing.
Madam, I'm working with the tissue culture of date palms. The problem I face with it is the browning of explant preparation and culture.
See, some of the plants, they have relatively high concentration of phenolic compounds. Now these plants, whenever they are injured, that is what is done when we prepare an explant, they are cut into pieces. So when they are injured, they liberate a lot of phenolases. These phenolases oxidize phenolic compounds, resulting into formation of a brown inhibitory compounds, and they cause the browning of either explants. Now, phenolic compound formation need not be due to injury only. Even in general growth processes also, they are formed in excess, and that's what you see when you see browning of the callus or browning in vitro.
What are the steps that should be taken to prevent this browning?
Now, browning can be prevented by 2-3 different ways. One is by dipping the explants into antioxidants. Antioxidants that we use are ascorbic acid, cysteine, citric acid, glutathione, etc. All these are reducing compounds. So they are used, and taking a proper concentration, explant tissues are dipped into this solution for some time, and then they are inoculated into the media. Sometimes it is necessary to have these antioxidants incorporated into media also. For example, that's what you are doing, I believe, with your date palm cultures.
Madam, I am working on problems of tissue culture related to plants like groundnuts and orchids, and sometimes I get a condition in which the tissues appear to be glossy, and they look as if they are translucent and hyperhydric, and these plants don't grow properly.
Now, as you have said that they are hyperhydric, this hyperhydric condition is due to excess of water logging into the tissues, and because of that many physiological disorders arise and these tissues fail to grow normally. To solve this problem, here is a graph that I have presented, and that tells you what are the causes that gives this problem and how it could be solved. It can be solved due to by the temperature, or by altering agar concentrations, or by reducing ammonium concentrations, etc.
I am working on papaya. I have found some explants can grow in ordinary basal media, whereas other explants requires hormones at low concentration, but for some tissue required higher concentration. Why is it is so?
There is a condition called spontaneous hormonal autonomy. That means there are certain tissues which have inherently enough hormones to initiate growth, and after that synthesize enough hormones to continue their growth. Whereas there are other tissues which need external supply of hormones. Those which have hormones into them, such tissues are called habituated. For example, in case of tobacco, it was found that cortical cells are habituated. In absence of cytokinins, they can grow into a plantlet. Whereas pith tissues, they cannot grow unless external cytokinin supply is given. Same plant, but if you take leaf tissues, if you don't give them hormones, their growth will be very slow. But if you give them cytokinin, their growth will be faster. That means they are partially habituated. So habituation is a physiological disorder, physiological condition in which a hormonal supplement is required. This habituation is not a mutational character. because a plant that is grown from a non-habituated callus can also become habituated.
What is the difference between habituation and other physiological problems or disorders?
See, habituation is concerned only with the internal inherent level of hormones, whereas physiological disorders, of course, they include hormonal requirements, hormonal levels, but they also include other growth and requirements like food, nutritional requirements, growth patterns, or complex addendums like requirement for, say, casein hydrolysate or coconut milk, etc. Also physical conditions like temperature and their impact on the growth. Say for example, we give dark conditions during the initial phases of tissue culture, whereas we give them a known amount of photoperiod and light intensity during other phases. So those are the physiological requirements, and if there is any variation, then we come across certain problems, and those problems are called physiological problems. So habituational problems are related only to hormones, that to internal level of hormones, whereas physiological problems includes food, nutrition, growth, complex addendum requirements, physical condition, explant stage, internal growth, part of anatomy, etc., etc.
I'm working on developing a saline-resistant variety of groundnuts. Sometimes we fail to culture a plant from a particular tissue and also come across a block of non-totipotency. Why is it so when a plant tissue has been accepted to be totipotent?
See, there is no evidence to show that there is any chromosomal constituent which is responsible for totipotency. That we haven't discovered yet. And therefore, the block of totipotency, as you mentioned, that we face, is it really due to a non-totipotency, or is it because there is a lack of understanding regarding the requirement of nutrition at that particular phase of growth of that tissue? We don't know that, and therefore totipotency or non-totipotency still remains a problem to us. Any morphogenetic failure into development need not be due to non-totipotency.
What are the various causes of genetic variability in culture?
See, like in nature we get polyploidy or aneuploidy. In cultures also we get ploidy, and often we get polyteny also. Polyteny is endomitosis and then reduplication of chromosomes. And due to this polyploidy or aneuploidy or polyteny, we often get somoclonal variants formed in our cultures.
Madam, what are somoclonal variants?
Somoclonal variants are variants formed due to alteration of chromosomes or chromosomal characters in somatic cells, and often they are non-nuclear in origin, and that's why we call them somoclonal variants. This is a culture of an orchid. Here you can see that the original protocombs that were forming were dark green, but over a period of months subculture, we started getting light yellow protocombs. These light yellow protocombs eventually developed into a plantlet which had some morphogenetic differences in their leaf color in the plants.
Are mutational events in tissue culture always of nuclear origin?
No, it occurs at mitochondrial genome also. Mutation can take place in mitochondrial genome as well as in chloroplast genomes.
Should we then believe that there will be absence of uniformity?
See, absence of uniformity is due to frequent occurring of somoclonal variants or seasonal variants in the cultures. But there has been enough evidence of getting genetically stable plants in tissue cultured orchids. For example, in Phalaenopsis, in Dendrobium, in Cattleya, etc. For years together, they have been subcultured and tissue cultured, and they have been getting, of course there has been some reports of getting variegated leaves, etc. But they have also added or gone in favor of advantages because they produced more beautiful looking leaves also along with the flowers. But stable plants have been achieved by tissue culture. So we cannot go on the continuous belief that a tissue culture plant always ends up into variants.
Madam, what is the epigenetic control?
See, explants from the different parts of the plants respond differently to hormones and produce different organs, even if they are subcultured. This type of behavior is ascribed to epigenetic control. Epigenetic control need not be nutritional or hormonal. It can be due to physical factors also. For example, if we take embryogenic callus, there are certain callus, if you give them say 22 degrees centigrade, they will form embryos. But if you give them 32 degrees centigrade, they will fail to form embryos. This type of characters are also epigenetic traits.
What is their significance in tissue culture and can they be helpful?
Well, let me first tell you what is an epigenetic trait is. Epigenetic trait is defined as the one which is stable throughout the number of cell generations and in the absence of inducing factors. For example, salinity resistance developed may be due to involvement of epigenetic determinants rather than mutational in origin. And epigenetic trait need not be always a hindrance to micropropagation. They often help in getting better varieties, improved varieties also. So in this group discussion, we have discussed about some of the general problems that we encounter during tissue culture. For example, problems which were of chemical nature and problems which were of biological natures and problems related to physical factors. And these problems were like browning of the explant or callus, vitrification of the cultured material, habituation of the explants and the growing callus or tissues, non-totipotency, physiological disorders, genetic variations, absence of uniformity, and epigenetic traits and their control over our cultured products. We don't say there is a permanent solution to these problems, that we can do certain things and these problems will not occur or reoccur in our cultures. They will keep on occurring, but as they come, we have to combat them.